Okay, I think we're gonna get going. People can come in as they want. Welcome to our June Tech Talk. If you have any questions, please put them in the chat and Reiner will answer all your questions at the end of the talk.
Yes. Okay, thank you. So welcome to the June Tech Talk from Nuance Center, and my name is Reiner Blair, and I want to talk a bit about cryogenic sample preparation for electron microscopy, considerations and techniques. So my first slide, when it ever comes. Okay, so I want to start with a slide that shows some differences between samples that are cryofixed here on the left side and samples that are, which is usually by plunge freezing or high pressure freezing, and samples that are processed conventionally on the right side. So the goal for both procedures is to retain the ultrastructure of the samples as unaltered as possible, of course, and to allow us to study the samples in the high vacuum conditions in the electron microscope. With cryogenic fixation, smaller samples in suspensions like protein molecules, virus particles, or liposomes are plunge frozen, while bulkier samples like tissue pieces or hydrogels are high pressure frozen. The purpose of both methods here is to freeze the samples without the formation of ice crystals. If this goal is achieved, then the frozen samples are as close to the native state as possible. Conventional processing at ambient temperature includes chemical fixation and dehydration with organic solvents, then resin embedment for TEM, or it consists of dehydration followed by critical point drying for SEM. Each of these processes can induce changes or artifacts in the samples. For example, permeability changes lead to redistribution and or loss of diffusible ions, and small molecules can also be lost or extracted like lipids. Also, we can observe shrinkage, distortion, or collapsing of smaller samples during conventional processing. An example is shown here. So this is a piece of a hydrogel. The left side shows the hydrated sample before conventional processing. And the slice here is about 3 millimeter by 3 millimeters. And we can see that after conventional processing with dehydration and critical point drying, it shrunk to a piece of about 2 millimeters by 2 millimeters. And it shows also signs of distortion. So I think this is quite striking for demonstrating the effects that conventional preparation can have on samples. But as mentioned before, to take advantage of cryogenic sample preparation, we need to be able to freeze samples without ice crystal damage. And there we go to the next slide. So here we see that ice crystal formation is a main challenge because water is the main component in biological specimens. Usually it's more than 50%. And also liquid water is the most mobile component in biological systems. The main task of cryofixation, therefore, is to keep the water at the same place it occupies at room temperature. And this requires either very high cooling rates or the application of pressure during freezing. So which cooling rates can be and have to be achieved, which is seen in this figure here on the left. From a theoretical point of view, cooling rates are limited by three factors: by the heat capacity and the heat conductivity of water, and by the heat flow that can be achieved at the specimen surface. And we can only influence this last parameter, which is the heat flow. Now, calculations show that the highest possible freezing rate drops drastically with increasing distance from the cooled surface. At a depth of 0.1 micrometers, a cooling rate of 10 to the 7th degrees per second may be attained, as we can see here. But at 10 micrometers distance from the sample surface, only about 10 to the 5th degrees per second, and at a further distance of 1000 micrometers from the surface, the cooling rate goes down to about 10 degrees per second, which is extremely slow. Practice has shown, on the one hand, that sufficiently high values can be attained, and on the other hand, that the theoretically predicted limitations are correct. So pure water needs more than 1 million degree per second freezing rate, and it has been possible to vitrify water in a thin layer. I mean, we are doing this on a regular basis in the Vitrobot, more or less. Although that is not pure water, usually it's a physiological solution. So the graph on this shows also that that 1 is the range of vitrified pure water, 2 is the range of vitrified animal cells and tissues, the Roman numbers, and 3 is and 3 is the range of specimens that are vitrified under high pressure. The cooling rate drops drastically with increasing distance from the cooled surface. So cells and tissues need cooling rates in the 10,000s to 100 to up to 100,000 degrees per second, and they can be cryofixed in a superficial layer up to 10 micrometers in thickness. So we have certainly limitations here. It is interesting that the advantage of double-sided cooling is limited. In this context, the question arises and has to be answered. In the 100 degree per second range, double-sided cooling enlarges the thickness of good cryofixation, and we should expect the same phenomenon in the 10,000 degree per second range. That is, instead of 10 to 20 micrometers, we should be able to vitrify specimen sandwiches about 16 micrometers thick. Our experience has shown that this has never been achieved, neither by plunge nor by jet freezing. And why do we have this discrepancy? The problem is that both sides with double-sided freezing need to be frozen simultaneously, and if we have a freezing rate from 10,000 degrees per second, we also need simultaneously freezing in the 1/10,000th of a second, and this is basically not achievable. So this can be achieved only when we have lower freezing rates, like is the case, for example, when we freeze with the help of pressure. When we apply pressure, we only need pretty slow freezing rates in the hundreds of degrees per second, and in this regime, we are actually able to obtain double-sided freezing and have a positive effect of it. So now that we have seen that we need freezing rates of close to 100,000 degrees per second to vitrify biological samples or samples with over 50% water content by plunge freezing, we can see from this table that Freon 13, propane, and ethane are good candidates as cryogens. However, Freon 13 is a greenhouse gas and was more or less forbidden in the 90s, and so that cannot be used anymore. But with ethane and propane, we can achieve freezing rates of close to 100 degrees per second, and this makes them suitable for plunge freezing. It's also clear from this table that nitrogen is not a very good coolant, because we have extremely slow freezing rates because of the Leidenfrost effect. So the next slide shows different approaches to achieve different freezing thicknesses. We have the plunge freezer, which freezes about to 10 micrometers. We have the spray freezer, which can achieve about 15 micrometers of sample depth that is vitrified. Same is true for the slam freezer, and the propane jet can go up to 40 micrometers, while the high pressure freezer achieves the highest thickness with around 500 micrometers. But please keep in mind, those are ballpark numbers, and of course there are variations depending on samples, of course. So with this, I want to focus on the two main freezing methods that we use for cryogenic sample preparation in our lab. That is plunge freezing and that is high pressure freezing. And the most straightforward method for cryogenic sample prep is plunge freezing. It's performed at ambient temperature, and it is a fast technique. The sample is applied to a substrate, usually a TM grid, and frozen in LN2, liquid nitrogen. Once frozen, the sample can be observed with cryo-TEM, cryo-SEM, or cryo-STEM. However, the method is limited to very thin samples only, as we have seen, that do not exceed a thickness of a few micrometers. Up to 10 micrometers are possible. And it's the method of choice for small samples in suspensions like molecules, virus particles, liposomes that can easily be prepared within these thickness limitations. There is, of course, overlap between methods, so we could freeze dry samples that were plunge frozen and study them at ambient temperature, for example. Also, we could use cryoprotectants and plunge or flash freeze thicker samples for particular applications. Sorry? Okay, for example, for immunolabeling, as mentioned here on this slide. In particular, at BioCryo, we often have non-biological samples that we treat with cryoprotectants and that we plunge or flash freeze and then process more like high-pressure frozen samples. That is why I place a faint plunge freezing here on top of the high-pressure freezing. It's also important to note that the samples prepared are not restricted to electron probe-based studies or instruments. We can also use them for light microscopy, for AFM, for laser ablation, or for X-ray based methods. So in the next few slides, I want to focus on the plunge freezing process. So plunge freezing is performed with an automated device. At BioCryo, we use the VitroBot Mark IV in our Hogan lab and the VitroBot Mark III in the AB-Wing space. The purpose of this instrument is to produce vitrified thin films of samples on TEM grids that can be used for cryo-TEM, and also to do this in a reproducible manner. In the first step, a grid is mounted onto special tweezers that are then mounted onto the VitroBot. The grid is then lifted into the specimen chamber. A few microliters of sample solutions are applied through a side port. Excess liquid is blotted off, and the tip of the tweezers with the grid and a thin film of sample solution is rapidly ejected into liquid ethane in a cup that is cooled with liquid nitrogen. The bottom image shows the styrofoam cup that contains the liquid nitrogen bath that cools down the metal cup with liquid ethane in the center. The frozen grid is then transferred into a pre-cooled workstation with a pre-cooled cryo transfer holder, and the cryo transfer holder is inserted into a TEM for observation in cryo mode. So, and during transfer from ethane to the cryo transfer holder, the grid is always kept under liquid nitrogen. And the cryo transfer holder is cooled with liquid nitrogen in its dewar. It can also be connected to a controller for monitoring or adjusting the temperature of the tip during observation. So there are several parameters that influence plunge freezing. So we have, for example, the choice of grids. There are different kinds of TM grids that can be used. For example, lazy carbon, quantifoil grids, or grids with regular arrays of open areas, grids with continuous carbon films, as well as grids without any support film. Also, usually grids are pre-treated with plasma to make them hydrophilic before samples are applied. And it is also advised that the ethane should be as cold as possible for freezing a sample, and this is indicated by the presence of frozen ethane around the inner perimeter of the ethane cup, which is shown in the picture you can see here. So this is the frozen ethane. And there are also, of course, several parameters on the VitroBot itself that need to be set properly. Temperature and humidity in the specimen chamber, as well as blot time, the number of blots, and the drain time, if any, for example. The table lists a few examples for different settings. Another factor is, of course, the volume of the sample that is applied. And since we are operating a Mark III and a Mark IV Vitrobot, I want to point out the differences between these two models on the next slide. So we can see in the console menu for the Mark III here on the left and the Mark IV, everything is the same. So we can adjust temperature, humidity, and there is a timer which nobody ever uses. We also have the same place new grids, start process, and plot reset plot papers menu points. And also for the options menu, we have basically the same points with plot time, total number of plots, wait time, drain time. It's just that it's a bit differently arranged, but the difference between the both is here. So in the Mark III, we have a plot offset parameter, and that regulates the vertical position of the grid between filter pads, while in the Mark IV, we have the plot force, and the plot force regulates the distance between the two filter pads. And this influences the force that presses the filter pad against the grid during plotting. So in any case, for consistent results, it's very important that a grid is always taken up identically with a tweezer, because obviously if the position of the grid is different between different runs, it makes no sense to compare any parameters. Okay, then and in an overview, we can divide the whole plunge freezing process into 4 main stages. So we have sample optimization, and we have sample carriers and treatments. We have deposition of the sample, and we have vitrification. So each of the stages has several points, options that can be adjusted, and the most options are available for sample optimization, of course, like the design of the sample in the first place, the buffer pH, salt conditions, additives, detergents. So there's an endless variety, of course, that can influence the sample. For the carriers and treatments, we have, of course, a lot of options. We can use different grids, as I mentioned before, with carbon films or foils, continuous film. We can have chemical treatment of the grid surface. And yeah, we can have plasma treatment, of course, or cleaning. And of course, deposition is something where we have not a great variety of options here with our Vitrobot. Obviously, we deposit the sample with a pipetter, and the sample is then blotted to remove excess fluid before freezing. But there are other techniques available on the market or emerging, which are droplet-based methods or scribing methods. But I will talk about one of those methods later briefly. But at the moment, we only have this blotting option. And of course, with vitrifications, we are doing the plunging, but in theory, there's also, or not in theory, but there's also jetting available. So the choice of grids, it can be very important. And my impression is that most users rely on lazy carbon grids, which is absolutely fine. But I just want to make sure that you have an idea of the options that exist. Beside lazy carbon and quantifoil grids, for example, there are C-flat grids with carbon films that are extremely clean and flat, of course. And there are gold foil grids that are very stable in the beam and can help if movement or drift of carbon films is an issue. So if parameters are not optimized in plunge freezing, artifacts might be introduced, and here are a few examples. So the graph shows the rate of evaporation of a thin water film at several temperatures and environmental humidities. We can see that at room temperature, which is this curve here, evaporation rates are high. So 100% humidity is almost in every case required to avoid sample damage. And the image below the graph shows an example for osmotic collapse of spherical liposomes into vase-like structures. And in this case, approximately 50% of the internal volume of the liposomes was lost during preparation at 25 degrees and 40% relative humidity. So further artifacts that we can observe sometimes on the right side shows, for example, water contamination from water vapor pressure in the column of the electron microscope. So this usually can be easily removed by irradiation of the sample. So E here is before the electron beam is put on the sample. In F, we can see here an area, this area was irradiated with the electron beam and this not. So here we still have this artifact while here it's gone. And here we can see here we can see this circle. The brighter area is the area that was actually exposed to the electron beam. G here on the side shows a hole that was burned into a vesicle by overexposure or by electron beam damage. Some more plunge freezing artifacts, for example, we can see A is a freeze drying effect in a film because the sample had a bad contact with the holder tip and was warming up. And then in the vacuum of the electron microscope, we have a freeze drying effect. B shows a thick but intact vitrified film and partially damaged or ice-contaminated vitrified film squares on a quantifoil polycarbon grid. C shows severe electron beam damage, bubbling at the rims of cubic nanoparticles. D shows ethane residues from vitrifying procedure on a film of cubic nanoparticles. E shows hexagonal and cubic ice structures because of heating up of the sample with the electron beam. And F shows a segregation of particles in a film of the emulsion, emulsion of particles. So we can see that larger particles are located in different areas as smaller particles, which is segregation. So with this, I want to switch to just one new development in the plunge freezing area. And for example, it has been shown that the time between the application of a sample to a grid and the freezing should be kept as short as possible. The figure shows that as soon as a sample is applied to a grid, interactions with the air-water interface and sample occur, especially in the case of smaller samples, like for single particle analysis. And it's also shown that the distribution of samples changes rapidly with time. So the difference here is 500 milliseconds and 170 milliseconds between applying the sample and freezing. And this led to the idea that only such a small amount of sample should be applied to a grid that no blotting step is necessary. And for this purpose, a self-plotting grid was developed. It is a mesh grid covered with a patterned film, and the surface of the grid bars is covered with nanowires, and these nanowires provide the plotting function. So this self-plotting grid is unfortunately not intended for use with the VitroBot, but with a special instrument named a Chameleon. And this instrument is a fully automated device that takes grids up automatically. It has an integrated glow discharge unit, and only a few picoliters of sample is automatically dispensed onto the grid. The nanowire grids make plotting obsolete, and the grid is frozen immediately. So after freezing, the quality of the frozen sample is evaluated with a camera. Before then it's decided if the grid is kept or if it's discarded. But so this new device has so far only been used with samples for single particle analysis, and there is not really a usage with larger samples so far. But there is a chance that this device would also work with larger samples, and this might make it attractive to you. Just wanted to give you an idea about new developments here. So with this, I want to switch from plunge freezing to high pressure freezing. And high pressure freezing allows us to vitrify larger sample volumes with thicknesses up to 500 microns. The limit depends mainly on the water content of the sample. And there are several workflows that can be followed with high pressure freezing. So we can have, of course, we can freeze dry the sample and observe the sample with SEM. We can freeze fracture, etch coat the sample for cryo-SEM. We can do freeze substitution, which ends in a resin embedded sample, and that can be used for TEM, STEM, and SEM. And of course, we can do cryo-ultramicrotomy or cryoplaning, and then the sample can be observed with cryo-TEM, cryo-TEM, or cryo-STEM. So the main path at BioCryo with high pressure frozen samples is freeze substitution. So this route here, and this is the route I want to explain a bit more in detail now for the rest of the talk. And again, it's also always important to keep in mind that those samples are not just for electron microscopy, but for many other applications too. So we have seen the limitations in sample thickness that apply for the plunge freezing technique. There is one way to overcome this hurdle, and that is the application of pressure to samples while they are being frozen. This approach is based on the well-known fact that when water freezes, its volume increases. This is based on Chatelier's principle. And this states changes in the temperature, pressure, volume, or concentration of a system will result in predictable and opposing changes in the system in order to achieve a new equilibrium state. So that means by applying high pressure during freezing, we achieve two effects. First, the volume expansion is prevented, and we prevent the formation of ice crystals. No formation of ice crystals means that less heat is produced by crystallization and less heat has to be extracted per unit time by cooling, which results in a reduction of the cooling rate that is required for vitrification in this case. And how is this achieved? Well, high pressure is lowering the freezing point by a reduction in the rates of nucleation and ice crystal growth, and the optimal range of pressure can be deduced from the phase diagram of water that we see on the right. And we see from that the most profitable pressure zones is just above 2050 bars. And at this point, the melting point of water is lowered to a minimum of minus 22 degrees. And the zone of supercooling is expanded down to minus 90 degrees Celsius. What we also need to consider is the temperature ranges of water crystallization as shown in the table. So this is very important since the range in which freezing damage by crystallization can take place is limited to the temperature zone between the melting point and the devitrification temperature of a system, as we can see in this table. When the devitrification temperature is attained, viscosity reaches a high value, and this prevents molecule movement and consequently suppresses ice crystal growth. This crystallization range between the melting point and the devitrification temperature is related to the water content of biological objects. So in physiologically active cells and tissues, the temperature interval may extend from minus 2 degrees to minus 80 degrees. There are also more frost-hardy samples where the range is between minus 13 and minus 43 degrees only. For pure water, to prevent the degradation of water in frozen samples, the samples need to be kept always below the devitrification point of water to be on the safe side, which is around minus 133 degrees Celsius. So this image shows an example of a sample that was just frozen in liquid nitrogen without any high pressure or without any cryoprotectants. And we can see very well, so it's a brain slice of 300 micrometers thickness, and we can see that the structure is heavily damaged by hundreds of those white gaps that were produced by expanding ice crystals during freezing. So this is a free substituted sample, and we are looking here at a resin section of this sample. So here we see the HPM 100 high pressure freezer and the diagram of a typical freeze on the right side. So the time scale of the whole graph is 20 milliseconds. And we can see that a pressure of over 2000 bar is reached after about 4 milliseconds, while the temperature drops to minus 100 degrees, which is the orange line, in less than 10 milliseconds. So that's the temperature drop here. This is a cooling rate well above the limit to prevent crystallization of water in biological samples. And in fact, as we have seen before in the table of water crystallization ranges for biological samples, the devitrification temperature is usually around minus 80 degrees, depending on the water on the water content. And so this means high pressure freezing is really a very effective method for freezing larger samples without any crystal damage. So while we can freeze larger sample volumes with a high pressure freezer, there are limitations to sample size. So here we see the three basic sample holder types that can be used for high pressure freezing. We have the 3 millimeter Plunge system, we have the 6 millimeter system, and we have the tube system in which small copper capillaries are frozen. So the flat system here with the planchettes can be used for tissue pieces. Of course, it can also be filled with cell suspensions, and liquid samples in general can be drawn into those capillaries and frozen. This is also very good for cell suspensions or samples in general that are suspended in solution. And one approach for sample prep that we do successfully is we make slices of our sample of a defined thickness with our VT1200S vibrating plate microtome. And we then use tissue punches, as you can see here, to cut out round discs of those tissue slices that fit perfectly into those carriers. And this slide shows several variations, how we can use those flat carriers, for example. They are available with different depths on each side, and we can combine them to form sandwiches that contain our sample, and those volumes can be adjusted by combining the respective carriers. We can also use a sapphire disc with a monolayer of cells and a spacer ring and freeze those. Or here we can see the copper capillaries. In any case, it's very important to keep the processing time as short as possible, since we want our sample to be in a very good, healthy condition when we high pressure freeze. And we have to avoid air bubbles at all costs, because air bubbles would be compressed by the pressure and they would delay the buildup of pressure. And our sample would show probably a freezing damage if we had bubbles in it. And for example, if we use a layer of cells or so, we use space fillers. So we can use hexadecane, dextrane, BSA, or yeast paste. That depends, of course, on the sample. And we also have the options, if we have smaller samples, we can, like single mouse egg cells, what we use sometimes, we can use them. We can put those into cellulose capillaries and then put those cellulose capillaries into the copper capillaries for freezing. So this slide here shows again how the whole assembly is formed. So we have the carriers that contain the sample. We have a mid plate here, and we have the two cartridges. So, and usually a sandwich is formed with the two carriers. It's placed in the mid plate, and this mid plate is then placed on top of a cartridge half, as we see here, and then the other cartridge is placed on top for freezing. For the copper capillaries, we have special cartridges in which the copper capillary is placed. And here we see a short sequence. So we can see here a carrier that contains a sample, and I will put the lid on top of this carrier. That one has a flat bottom, and then the sandwich here, the cartridge system is built, and the sample is frozen. It's a very short sequence. So, and with this freeze, the sample is dropped into a dewar with liquid nitrogen. And from this stage on, we can either store the sample for further processing, or we can of course continue right away. And the main route, as I mentioned before, that we process our samples after they are high pressure frozen is freeze substitution. And freeze substitution is a process in which we replace the frozen water of the sample with an organic solvent at low temperature. And in this way, we avoid damaging effects that are usually caused by dehydration at ambient temperature. So we have a special device for that. So it's our automatic freeze substitution device, AFS2. And usually the procedure goes like following. So we program all the single steps on this instrument. The sample is then placed in the chamber here. The chamber is already pre-cooled. So this is a liquid nitrogen dewar that keeps the temperature. And then usually those protocols are that the sample is kept at -90 or -85 degrees for several days. And during that time, the water is replaced with a freeze substitution medium. Freeze substitution medium usually is acetone with glutaraldehyde or uranyl acetate or osmium tetroxide. Sometimes it can be a combination of those. And then the temperature, after several days, the temperature is then gradually ramped up until we are at room temperature, and then we can take the sample out, do some rinsing steps, and do the resin embedment procedure. So this is one way. Depending on what we want to do with our samples, we can also end the run at low temperature, keep the sample at -50 or -40 or -20 degrees, and then infiltrate the sample at these low temperatures with resin. And those resins then also can be polymerized with UV light at those low temperatures. And this can be very helpful for samples that we want to use for immunolabeling, or for samples that we want to retain with fluorescence. So samples that have been labeled with fluorescent markers and then be frozen. And if we follow those procedures, there is a chance that we can make slices that can be observed then in the light microscope with fluorescence before they are observed in the electron microscope, which would then be a correlative approach. A few examples are shown here for freeze substitution. So we have, this was produced not too long ago in our lab, frozen. And we can see how well synaptic vesicles are retained in these neurons from C. elegans. It has been shown that the density and uniformity of synaptic vesicles and the size of extracellular space differs in neuronal or brain samples between high pressure frozen and conventionally prepared samples. So on the right now we see two other classic samples from the literature. One is the left one here is from a marine sponge. We are looking at the same areas in a sample, extracellular space, and Golgi apparatus is seen here, and the conventionally prepared sample, and we look at the comparable area with the high pressure frozen and freeze substituted sample. It's clear that we can see the extracellular content much better. It's not extracted or destroyed, and also the Golgi apparatus and the cytoplasmic vesicles in general are much better preserved. And the right side here is a bacteria sample. Here the upper one is a high pressure frozen freeze substituted, while the lower one shows conventionally prepared samples, and the differences between those samples are quite obvious. So with this, I want to switch briefly to a different example. So comparison between conventional SEM and cryo-SEM of hydrogels. So here we see a hydrogel that was processed conventionally with dehydration and critical point drying and then observed with SEM. And here we see a cryogel sample that was high pressure frozen and freeze fractured and then observed with cryo-SEM. And we can see that the conventionally processed sample has many collapsed areas and collapsed fibers, while the sample that was high pressure frozen and observed with cryo-SEM looks way more or less damaged by the processing procedure. And one last example, so we can use those samples for really a lot of applications. For example, here we see a sample. This is a copper capillary that was high pressure frozen. We are looking into the chamber of a cryo ultramicrotome. The sample sits here. The diamond knife is here, and we were using silicon nitride windows to put cryo sections on top of them. And those silicon nitride windows were then used at Argonne National Lab for examination of elemental content with X-ray based methods. So this is still in process. So with this, I want to stop. Thank you for your extension, and I'm ready now for taking your questions.
If you have any questions, you can either put them in the chat or feel free to unmute yourself.
Rainer, that was great. This is Hank Seifert down in Chicago. There's so many different ways to prep for this. How do you figure out what to use on what?
Yeah, I mean, that is always, that's very important indeed. And that is why we usually always have to talk with users to find out what they exactly want in order to decide which method is best. Because it's certainly not always, it does not make always sense to start with the most complicated technique.
So you start with the simplest, see if it works, and then sort of go up in complication if you need to.
Yes, I mean, if someone wants to look at very difficult to retain structures, of course, it's clear we have to freeze the sample. And if the sample exceeds a certain thickness, then we have no choice. We have to do high pressure freezing. An option would be to try cryoprotectants, but they often influence the sample also, of course. So it really always depends.
Thank you.
Thank you.
Hi, Rainer. Great talk. Thanks. I was just wondering what cryo freeze substitution method you apply the most and why.
I mean, there are standard protocols, of course. So for best structural preservation, just for looking at the ultrastructure, usually we use osmium tetroxide in acetone. And the protocol is you keep the sample at minus 95 or 90 degrees for 4 to 5 days, and then you go up with 5 degrees per hour until you reach room temperature, and then you can do the resin embedding at room temperature. So that is, I think, is the most the standard approach for high pressure frozen samples and free substitution.
Okay.
But as I mentioned, if you want to do immunolabeling, you have to do everything to retain the hydrate shell around proteins and to avoid denaturing steps. And that is why you want, in this case, you would want to keep the sample always at very low temperature. And you would use a low temperature resin for embedment of the samples.
Thanks.
So any more questions?
All right, well, thank you for attending
Okay.
our Tech Talk. If you have any questions you think of later, feel free to reach out to Reiner. His contact information is on our website under BioCryo. And we'll be posting a copy of this PowerPoint on our website as well.
Yes.
Thank you.
Okay, thank you all for attending.